tell he's the boss. Okay, so the next speaker is David Gayton. It's a super pleasure to have him here. He's a new faculty member in the Department of Neurology. Has a substantial and interest in the immune contributions to neurodegenerative diseases. He's really a pioneer in this field, and he's doing really, really, really exciting work. And David, please come up and tell your story.
Okay, thank you, Dr. Kaub, and thank you, Dr. Osdendler, and congratulations on year number 14 of this wonderful symposium. I've had the pleasure of being in here as an attendee in the past, but it's wonderful to be able to share some of my laboratory's research with you all. And it's rare that I get the opportunity to present to scientists and foundation leadership, and also families, caretakers, and patients alike. So that gives me the opportunity to thank the entire ALS community for everything that we're doing together. I have to say I'm relatively new to ALS research. I came to Northwestern 3 years ago. My background is in Alzheimer's disease. But what I noticed looking at the field of ALS is that there's really, compared to Alzheimer's disease, a noticeable lack of knowledge around the immune system in ALS. And so I'm going to share with you some data from my research lab. I want to say also that we only study humans, so we study human cells and tissues, which is another reason why I'd like to thank those of you who participate in these research studies that we are so fortunate to do. So as Bob alluded to, I study the immune system. You can call me a neuroimmunologist. And my laboratory uses genomics tools, and I'll talk a little bit about what these tools are and how we use them to study the immune system in ALS. So this study started with an analysis of the peripheral immune system. So these are blood samples that we drew from healthy people. These were generally caretakers or family members of patients with ALS. We had 18 healthy controls and 22 patients with ALS. And we extract out the immune cells from their blood, and we sequence them. So we analyze their genomic code. And what we're interested in is how the genomic code of cells of the immune system in ALS patients might be different from that of healthy people. Believe it or not, this really hasn't been done before with this type of sophisticated assay. And so these are characteristics of our patient population. It's important when you're studying the immune system that your study is age and also sex matched, and so we have that within this cohort. And so we sequence individual cells of the blood from these 40 people. And this is what our data looks like. Each individual dot here is an individual immune cell. So this is all the data from all of the samples that we collected. Altogether, it's about 300,000 cells in total. So now that we know all the different types of immune cells that we've got in our analysis here, we can start looking at what cell types within the immune system might be changing the most. And so each of these spots is colored by the type of immune cell that they correspond to. So when you look at the immune system of ALS patients compared to healthy people broadly, you don't really see many changes. If you just count the number of cells, this is what the data look like, where each individual bar is an individual patient. You can kind of see the data are scattered. There's really no clear pattern here. And when you sort of average them out by the different sort of groups that they correspond to, in this case, we have people who have slow progressing ALS and people who have fast progressing ALS. We also have patients with C9orf repeat expansions. There broadly does not appear to be a significant change in the shift of the numbers or types of immune cells in the blood. However, when we look at the genes that are changing, so this is a more up-close look at what these cell types, what their genomic material looks like, then we can start to see differences. And we can also distinguish different types of patients within this data set. So patients, for instance, with C9orf repeat expansions are very different from their healthy control counterparts. There are 273 unique genes that differentiate these groups. So now we can start to look at, okay, what are the immunologic changes corresponding to people with C9orf repeat expansions, and what are the cell types that those correspond to? And we see here that there's a CD14 type of monocyte. This is a cell that's important in your immune defense. So this is a cell type that in C9orf repeat expansion carriers is significantly different than healthy controls. And this is a cell type that now we can probably look at more closely in this specific patient group, perhaps to try to limit inflammation or adverse inflammatory responses in this patient group. We can also see differences in those people who proceed slowly or fast clinically with the disease. So we see immune cell changes that are popping out basically whichever way we compare these different groups to one another. And we can also look specifically at what those genes are, and we can ask ourselves, what are these genes corresponding to? Maybe there's a gene here that is already being targeted clinically to reduce inflammation, and we can repurpose this to treat these patients. Suffice it to say, we see a lot of inflammation in ALS patients, and we see inflammation in particular cell types called monocytes. As I said, these are a very important cell in your immune response. So that right away, we were starting to see that there's inflammatory responses in the blood of ALS patients, which was initially pretty striking to us. So we're fortunate to have a really excellent research community here focused on ALS. And we're lucky to collaborate with Evangelos Kiskinis, who's here also in the Department of Neurology. And Evangelos had a unique data set of sequencing of a large number of patients from various regions of their brain and spinal cord. And so we asked simply, can we look at this data set, which is much larger, comprised of more patients, and detect immune changes in this patient population as well? So here are the brain regions that we analyzed, and highlighted in dark red are the regions where we see the most changes corresponding to immune cells. And you can see the spinal cord here, which comes down from the brain, is significantly altered. So this became our area of focus. You can see highlighted in the red font here are various immune cell types that we saw were genomically changing with this assay, and they were specifically in the spinal cord. So this drew our attention to this region of the central nervous system for further exploration. We looked at immune cells with a marker called IBA1, which will label what we call innate immune cells. These are similar to those monocyte cells that I mentioned earlier. And you can see in a patient with C9orf72 repeat expansions, they had ALS. This is their spinal cord, and in white are the immune cells. And you can see here when we turn these immune cells into a red color so you can see them a little bit better. There's a high number of these cells in the ALS patient spinal cord compared to the healthy controls. So we next asked, can we get a closer look at these? Can we see what genes these immune cells are expressing in patient spinal cords? So we were fortunate to get a lot of patient spinal cords through a collaboration with Target ALS. And so they send us tissue on glass slides, and we can look at the mRNA. So this is messenger RNA that's encoded by your DNA and can reflect changes during disease. And we also look at a set of proteins in these tissues. So colored here are individual spots, so specific data points of unique patient spinal cords. So this data set has 15 unique spinal cords in it, and each little spot corresponds to a region of the spinal cord. And the neurons are generally found in this region here. Outside here would be white matter area. And so we use genomic assays and data science tools to convert that data into unique data points and look at what are the genes of the spinal cord that are distinguishing patients with ALS versus control counterparts. And again, we see a lot of inflammation. Now we're seeing inflammation in the tissue, in the spinal cord of deceased ALS patients. And we see a lot of inflammation in the white matter. This is where communication signals are sent through the central nervous system, is in the white matter. And to see this amount of changes and that they correspond, here I'm highlighting genes that are involved in inflammation. The fact that they correspond to inflammation to us signals that this is a response that's likely detrimental in the disease. You can also see a lot of changes in the anterior horns. This is where neurons can be found in ALS, or in the spinal cord generally. So we're seeing here in the anterior horns a lot of genes that are corresponding to an inflammatory response here in an area that harbors neurons. So now it's possible that we're seeing an inflammatory response that could be further damaging the all-important motor neurons in the ALS spinal cord. So finally, I mentioned that we can also analyze proteins. So we have a panel of immune proteins that we use in conjunction with our RNA analysis, and we can look at changes in immune proteins. So these are very important changes involved in the immune response, important proteins rather, involved in the immune response. So we see several of these immune proteins changing also in the white matter and in the anterior horns where the neurons reside. So we're seeing these important immune proteins corresponding to inflammation really highlighted here, showing in a particular immune gene called ITGAX. You can see it's much more high in the purple color in the patient with ALS versus the control. And that's true of these other proteins as well. So we also are interested in a type of cell called T cells. These are also an important component of your immune response. And we see a lot of these cells, interestingly, in the spinal cord. We're not sure exactly what they're doing, but we're interested in profiling these cells further with our tools. And to do something like that, we really need to apply sophisticated data science pipelines to our data. So we take data sets that have already been generated by others in the field, and we can apply them to our very important spinal cord tissues. So this is now single cell data that we can use to look at cell type changes in the spinal cords. So we take this information and apply it to our spatial transcriptomics data from the patient's spinal cords and ask, what are the cell types that are changing and what are the genes that are changing within those cell types? And this is what the data look like when you predict what cell type corresponds to each individual spot. So this is coloring of the spinal cord that we do ourselves, and then this is computational prediction of the cell type that's most likely to be found in that spot of the patient's spinal cord. So now we have cell type resolution. We now can look and ask, what are the cells that are changing? Are they immune cells? And if they're immune cells, where in the spinal cord might they be changing in ALS? And this image just shows you, using a marker called MAP2 to highlight neurons. These are labeled with a microscope image in green here, the nice overlap of that signal with our neuron signal using that predictive algorithm. So now we think we have really good idea of where the neurons are in the spinal cord, and now we can ask whether there's inflammation in those specific areas. And indeed there are. So now we can look at, in those individual spots where cell types are enriched, for instance, oligodendrocytes and neurons, are there immune system response genes found in those locations? And that is the case. Just highlighted here in yellow are some genes that are involved in the immune response. So these particular spots are enriched for oligodendrocytes, which are found in the white matter, and they generate a protein called myelin, which is important for your neurons to communicate. And then the second most altered cell type, of course, is neurons, and you can see in those spots enriched for neurons, we have inflammatory immune genes upregulated. So with these data, we can ask, what are the pathways involved in these inflammatory responses? Because now we're getting into the area of perhaps targeting these inflammatory responses therapeutically, if we can identify a pathway or a specific target involved in the disease. And so we see, for instance, TNF-alpha signaling via NF-kappa B. This is a very important pro-inflammatory immune response. Also complement, same thing there. It's a very important pathway involved in your immune response. So Dr. Kalb gave a really nice introduction on TDP43 pathology. And so with Evangelos's lab, we looked at areas with TDP pathology and asked whether there were immune cells changing in those areas. And indeed, that is the case. So this is an area where a TDP positive neuron would be found here. This is a TDP negative neuron, and the number of inflammatory cells is increased in the areas of TDP positivity, suggesting that these immune cells are reacting to the pathology. And finally, I'll leave you with this image, which is an immune cell of the brain labeled with this IBA1 protein, physically interacting with a TDP positive exclusion. So this suggests that the immune response could be against these misfolded proteins like Dr. Kolb discussed. I'd like to thank Charles Zhang and Lynn Van Os and Jake Bowles for their contributions to the work, and the rest of the lab managers who contributed, and I'd be happy to take any questions you may have. Thank you.
So if you would like to ask a question, but you can't walk to the microphone, please raise your hand. We can bring the microphone to you. In the meantime, may I ask the first question?
Of course.
So tying Dr. Kolb's presentation to yours, you talked about the neuroimmune modulation in ALS, and TDP pathology is one of the common problems with ALS, FTLD, and other neurodegenerative diseases. And neuroimmune modulation is also another common denominator.
Yeah.
How do you think those two play together? Do you think when there's TDP pathology, there's increased neuroimmune reaction, or increased neuroimmune reaction leads to worsening of the TDP pathology?
Yeah, I think the chicken or the egg question is important. I don't know the answer to that one, but I do think that TDP pathology could be immunogenic if loss of TDP from the nucleus is leading to production of peptides that otherwise wouldn't be produced if TDP was in the nucleus. Those could be immunostimulatory proteins. You had a question? Oh, hi. Hi, Senda.
Great talk. You talked about the chicken and the egg. Have you looked at the change of the immune profiling throughout the disease, like early versus late? Because we think that maybe it has a protective and then detrimental, or the other way around. The reason for that is none of the clinical trials that target the inflammation have showed any effect.
Yeah, I think this is an important thing to study, actually. I wish that we did have longitudinal samples to see, like you said, if it's a protective immune response early in the disease course versus late. We have patients who are progressing fast versus slow, which gives us some insight into that. But yeah, I'm not aware of any patient samples that have that longitudinal aspect.
Yeah, great talks. I was just wondering if you could quickly comment on CSF cells and also on you had C9. I was wondering if you had enough power in the peripheral cells to say something about SOD1 and FUS.
Yeah, we don't have any SOD1 or FUS mutation carriers in the data set. We actually see our biggest differences in C9orf patients, even though there's 4 patients in there, because it seems like C9orf has some kind of role in the innate immune response. It's really highly expressed by monocytes. As for CSF, this is kind of what I've always done, is analyze CSF. We don't have it at the moment where we're collecting CSF for research purposes from the ALS Center, but that's something that we're interested in doing.
We're working on it.
Yeah, we're working on it.